Lin and co-authors submitted a review to iMeta. The title is Gut X-axis. Here is the brief introduction of this paper. In this review, we applied the gut X-axis concept to describe the linkages between the gut and other organs, and discussed the latest findings related to the gut X-axis, including the underlying modulatory mechanisms and potential clinical intervention strategies. The highlights of this review include: the gut and gut microbiota modulate the homeostasis of extraintestinal organs. The gut microbiota-derived bioactive substances regulate the development of diseases in the extraintestinal organs. Targeting intestinal microbiota is a promising strategy for treating the diseases of extraintestinal organs. First, the gut-liver axis. The connection between liver and intestine is bidirectional. The intestine and gut microbiota may affect the progression of almost all types of liver disease. Second, the gut-kidney axis. Here is gut-kidney axis cross-talk communication mechanisms. Here is the intervention strategies based on the gut-kidney axis. The intestinal microbiological imbalance accelerates the loss of kidney functions. The homeostasis imbalance of the gut-kidney axis can also affect the normal functioning of other axes. Third, the gut-lung axis. The gut-lung axis is mediated by immune and endocrine pathways. Therapeutic strategies targeting the gut-lung axis. The gut-lung axis, a bidirectional interplay in health and disease. There exists a significant and intricate interplay between the gut and respiratory systems, with dysbiosis of the gut microbiota implicated in the etiology and progression of common respiratory diseases such as asthma, cough, lung cancer, and respiratory infections. Fourth, gut-heart axis. Gut microbiome metabolites, TMO, Pagan, SCFA, and MAVA, influence CVD development through various mechanisms. The development of CVD affects the composition of the gut microbiome, and in turn, the gut microbiome modulates CVD progression. Further research is needed to determine the effectiveness and safety of these interventions. Fifth, gut-bone axis. Potential mechanisms by which gut microbiota regulates bone metabolism. Gut microbiota and their metabolites influence bone health by affecting nutrient absorption, immunomodulation, the endocrine system, and extracellular vesicles. Sixth, gut-skin axis. Here is the crosstalk between gut and skin. Here is the links between skin diseases and the intestinal system. Skin health can be regulated by improving gut microbes and the consumption of probiotics and prebiotics for efficient repairing of the skin barrier. Targeting gut microbiota represents a novel approach to managing skin issues. Seventh, gut reproduction axis. The homeostasis of the gut microbiota plays a pivotal role in reproductive health by interacting with sex hormones like estrogen at every stage and level of female reproduction. Anomalies in the composition and functions of the microbiota contribute to the onset and progression of many reproductive diseases. Eighth, the gut endocrine axis. The gut endocrine axis involves complex interactions among the gut microbiota, enteroendocrine cells, and endocrine organs, playing an indispensable role in human health and disease. Regulating the gut microbiota through high-fiber diets and other methods to alleviate T2D has broad application prospects, but it is still in its early stages and faces numerous challenges before clinical application. Ninth, gut-brain axis. Here is the interaction pathway between the gut and brain in maternal and offspring host systems. Conceptual framework, mechanisms through which systemic interactions of maternal gut microbiota influence offspring's neurodevelopment. Maternal gut microbiota regulates offspring's neurodevelopment by influencing gut microbial colonization. Maternal gut microbiota affects the neurodevelopment of the offspring through the microbiota metabolic, immunity gut-brain axis. Herein, we summarize the possible pathways by which maternal gut microbiota regulates offspring's neurodevelopment from the perspectives of microbial colonization, gene expression, immunity, and metabolism.